All right, hello everybody. This next section that we're going to look at is photosynthesis. And remember, photosynthesis, there's going to be two parts. There's going to be a light reaction and the Kelvin cycle. So we're going to separate them into two nice parts, and then at the back end of it, we're going to look at the different types of pathways in terms of C4, C3, and CAM plants. Okay, so let's get started. Okay, so photosynthesis, the chapter that I need you to look at is on Moodle, chapter 1. It's all up on Moodle, so don't worry about that. Just bear in mind that whatever I speak, you have to know. So there might be some context in the Moodle chapter that might be like way over and you thinking, but Candice didn't talk about this. Then you just look around it, you look at the stuff that I did speak about. Is also, this is a very, very complicated section. It almost just punches you in the gut, and you go from kind of knowing what was going on at high school level to being what in this level. So I'm going to take it very slow. The bear, be proud of me. I've got to pace myself. This might end up being 3 sections of slides, and we're going to get through this step by step. All right. So the first thing you need to remember is that the plant is always going to be balancing this idea of carbon dioxide with water loss. Remember, we were talking about it in water relations, how the plant does a balancing act. And now you're going to see that this balancing act starts getting a little bit more intense. So you now have to, you're going to see, instead of just walking on a balance beam, you're now going to have to balance a cake on your head as well. So you're going to see now, okay, not only do we have to choose between whether we open the stomata and take in carbon dioxide, but do we risk the water loss? And do we risk having excess oxygen within our intercellular cell regions, intercellular air spaces within that mesophyll, to have a chance of running into Rubisco? What I'm saying should be utterly a mystery to you, but by the end of this module, by the end of this section, you should know exactly what I'm saying. So we're going to break this into the light reactions and the Kelvin cycle, and then we're going to have a look at the pathways, C4, C3, and CAM plants. But before we even start, I just want to quickly go back into leaf anatomy. I want to talk about this for a little bit, and I want to do some recap from high school. So we know that the leaf's job is to capture sunlight. We know that the leaf's main job is to photosynthesize. So the anatomy of this piece of equipment is been designed specifically to capture sunlight and convert it into energy. And it does this via a process of photosynthesis. So at the end of all of this, we're going to have the leaf capturing light. It's going to convert it into energy, and ultimately we're going to get carbohydrates or sugars or glucose from it. The structure of the leaf is what allows this mechanism to be able to do and handle photosynthesis. Now, you must understand the basic structure of the leaf anatomy, because when we start looking at C3, C4, and CAM plants, it becomes very important to understand the difference between a paleosade mesophyll and just a bundle sheath, or a spongy mesophyll. So just bear in mind, you will have chloroplasts in all of these cells, all the spongy mesophil, all the palisade cells. Remember that cuticle up on the top acts as a waxy layer. It's going to protect excess water loss. It's going to protect from too much sunlight. We have an epidermis layer, and then we have the palisade mesophils and the spongy mesophils. And you will see how the structure changes and how the bundle sheath changes. Bundle sheath is that yellow. The bundle sheath is this area over here that's covering the xylem and the phloem. And you will see how this will change between the C3 and the C4 pathways. But before we look at that, we need to understand the basic structure of a leaf. So we know photosynthesis is going to occur in these palisade mesophils, some in the spongy mesophils. Don't forget there's intercellular air spaces here to allow for carbon dioxide to come in through this says stomata. Don't get confused, it's stomata. Carbon dioxide is going to come in through the stomata and oxygen is going to come out. So basic. All right, so we know that this is specifically designed to handle photosynthesis. We've got chloroplasts in the mesophils, which is going to harvest the light, which is then going to create these reactions, the light reactions and the Calvin cycle, and ultimately produce glucose. The glucose is going to be transported by the phloem to the areas that it is needed. Oxygen is going to be released, and so on and so forth. So, quick recap. Basic recap. So remember, we discussed this idea of tropism. We discussed gravitropism in the first set of slides, and we know that this is the way it's going to move. And for photosynthesis to occur, for life to happen, we need light, especially for plants. And remember, tropism, phototropism, energy derived from the sun, and phototropism, the plant is going to move towards the direction of the sunlight. So if you are in desperate need of sunlight and you are in a shaded area, the chances are you're going to try and grow nice and tall to get your leaves exposed to direct sunlight, if that is what you are lacking, if that is what your Blackman's hypothesis is lacking. So remember, Blackman's hypothesis, you're only as good as your weakest link. In this case, if your weakest link is the sun, then you will have to grow up to try and increase that length to try and capture as much light as possible. So tropism, moving towards the sun, and the reason for this is the sun is going to be where we derive that energy needed to allow these processes, these biochemical processes to occur. Now remember, we harvest our energy. Now I'm talking about the energy that we switch the lights on and off of by fossil fuels. We also derive our energy. We are starting and we're moving to go onto renewable energy, such as solar power, hydropower, etc., wind power. We are moving towards sustainable energy, but plants have been doing this for thousands and thousands and thousands of years. They started doing it, and it is such, you can see the benefits of it, and you can see how sustainable it really is. And once we get to the nitty-gritties of photosynthesis, you're really gonna, I hope that you're really gonna be able to appreciate how in detail this is, and how almost perfect it is, and how it keeps on going, it's so renewable, it's so sustainable, and it's one of the very few processes in on this planet that is so renewable, that allows us continuation of energy from one form to the other form. Now, how do us as individuals get our energy? Because if I lie in the sun, I'm just going to get a sunburn. And I'm going to go red and blotchy, and then I'm going to peel, and it's not a pretty sight. So I can't derive my energy from the sun. How do I get my energy? I get my energy by eating plants and animals. Now, if you are a vegetarian, you take it straight from the plants. No hate there, only love. But ultimately, if you eat the plants, we are omnivores. If you are a vegetarian, you are just a herbivore. And we eat and we take the energy directly from the plants. Either way, your energy, whether you're a herbivore, omnivore, carnivore, cannibal, I hope you guys know what a cannibal is. If don't, Google it. Let me know on Discord. But yes, so however way, whatever you eat, ultimately you're taking it from plants. And it's the plants that are deriving that energy from light. So that is that famous equation that you can see there. 6 carbon dioxide plus 6 water molecules gives you 1 glucose molecule plus 6 oxygen. Now, that is the photosynthesis equation. That includes the light reaction, and that includes the Calvin cycle. So pretty much they've looked at both cycles together, and this is the summary. When you're looking at a paper, this is the abstract. This is the abstract of these two processes. It's by no means this simple. It gets a lot more complicated, and we are definitely going to have a look at that. So yes, you need to know this equation. Yes, you're going to have to produce this equation in a nice essay because it is the abstract of photosynthesis. But as you will see, it is not this simple. It is actually a lot more in detail. All right. Now, next slide. Remember, like I said, that most of the photosynthetic tissue of the plant is in the leaves, specifically in those mesophyll, the palisade mesophyll and the spongy mesophyll. In the mesophyll are cells, or in the mesophyll there's cells, and in a cell there's a chloroplast. Now, please don't get confused. Terminology is very important, and the terminology is going to be very important for the practical that is being derived from the section, which we will get to. But ultimately, from the section, you have palisade mesophyll, you have spongy mesophyll. In those mesophyll tissues, you have cells. In those cells, you have chloroplasts. In the chloroplasts are pigments that are green called chlorophyll. Why are the chlorophyll green? They are green because they reflect the green wavelength. So remember, we're going to get to all of this. They are green because they reflect the green wavelength and they absorb the blues and the reds. So when you see a plant that is red, it's reflecting the red. It is absorbing more of the greens and the blues. Okay, so what the color that you see around you is what that object is reflecting back to you. Hope that needs to be crystal clear. Okay, so we've got mesophyll. In the mesophyll, we have cells. In those cells, we have chloroplasts. In those chloroplasts, we have chlorophyll. And the chlorophyll is a pigment, and it is going to be this pigment that takes and absorbs the photons from light. All right, so the solar energy oxidizes water. Now, there's going to be two terminologies that we need to get clear because you're going to hear me saying them a lot, and that is oxidation or oxidizing or reducing. Oxidizing or reducing. You should remember it from chemistry, but the way I want you to remember it for photosynthesis is oxidizing. See that X, exit. It's taking. It's going to take it away from you. Whereas reducing, there's a D, you adding. Oxidizing, you taking it away from something. Reducing, you adding it to something. If you're an oxidizing reagent, you are the instrument that is taking it away. If you are a reducing agent, you are the instrument giving it away. Does that make sense? I hope it makes sense. It needs to be crystal clear. I'm going to say it again. If you are oxidizing, if you are oxidizing reagent, you are the agent that is taking it away. So what you're taking it away from is being oxidized. If you are a reducing agent, you are giving it, you are adding it on. If you are getting it back, if you are it's being given back to you, you are being reduced. So solar energy oxidizes water. It takes the electrons away from water, takes the electrons and it takes the hydrogen from water. It releases oxygen and it then goes off to the Kelvin cycle. It then reduces carbon dioxide by giving it more carbon compounds to form sugars. So this sentence over here, this first part over here is the light reaction in a nutshell. The second part, and down here, is the Kelvin cycle in a nutshell. Light reaction in a nutshell, Kelvin cycle in a nutshell. So solar energy comes in, it oxidizes water. Remember, the oxygen that we breathe comes from water and not carbon dioxide. We have elements that are released from the light system, which we will get to. It goes off to the Kelvin cycle. The Kelvin cycle reduces carbon, making it into a longer carbon compound, and eventually we get sugars, glucose. All right. Now, like I said, these are reactions that happen. So you've got in your stomata, we will get, sorry, not in your stomata, in your chloroplasts, you have thylakoids, which we will get to, and these thylakoid reactions, also called thylakoid reactions, don't get confused, is just reducing carbon dioxide to give us our carbohydrates, our sugar. This process of reducing carbon dioxide is called carbon fixation. Another name could be carbon assimilation. Please do not get confused. Don't get confused. They are one and the same. If you say reduction of carbon dioxide, it's the same as carbon fixation. It is the same as carbon assimilation. Okay. The thylakoid, in the thylakoid, we're going to get our light reactions happening. And so in the chloroplasts, we're going to get thylakoids. We're going to get a stack of them. It's called a granule. We're going to get to those. Don't worry. But in the thylakoid reaction, where that light reaction is going to happen, that first part of photosynthesis, the ultimate byproducts of that system is ATP, NADPH, and oxygen. That is very important. So the light reaction happens within that thylakoid region, otherwise known as thylakoid reactions. It's in the chloroplasts. We'll get to it. The main byproducts of it is ATP and NADPH, as well as oxygen, which is released back into the atmosphere. Those are then taken off to the carbon fixation reactions, which is in the Calvin cycle, Okay, and then it's made into your glucose. So these carbon fixation, the Calvin cycle, happens in the stroma of the chloroplasts. We'll look at a picture later and we will see it. In fact, let me just jump there quickly now. So here we go. You've got these beautiful thylakoids over here. You've got the stroma, the aqueous space around it, and then you've got a granum, which is a stack of thylakoids. In one of these thylakoids up at the top, you're going to have the light reaction. You're going to have that process where water's coming in. It's going to break down, and you're going to get ultimately out of that ATP, NADP, H, and oxygen. Oxygen is going to be released back into the atmosphere. The ATP and the NADPH is going to go to the stroma, and in the stroma is where we'll have the Calvin cycle, and the Calvin cycle will process to give us that glucose eventually. All right, so that's what this slide is. It's basically telling you very quickly, very briefly, what each stage is, how we're going to do it. So don't get confused. Thylakoid reactions, light reactions, don't get confused by the terminology. There's going to be different terminology used in different YouTube videos, in different textbooks. Just be aware, please. But the main crux from this is that your chloroplasts contain chlorophyll, and it's the chlorophyll that absorbs the photons. We've got this idea of light reactions and the Calvin cycle. The thylakoid reactions, which is the same term for light reaction, and then the synthetic process that occurs in the stroma, which is the Calvin cycle. I don't like that last point. I am going to delete that last point. Click save. All right, I'm happier. Okay, great. So now, waves. So now we understand basic, we understand everything kind of, we've got a basic idea. Now we need to run into this idea. Before we can continue, we need to understand the way light behaves. Now, you guys know from Chem 110, you know from school, all of that, that light can behave as either a wave or a particle. But ultimately, we're going to consider or look more to it as being a wave. But don't forget, particle and a wave, we're going to concentrate mainly on the wave part of light. Now we know that the light delivers energy in the form of photons to plants, and only some of the photons absorbed are used by plants. Remember, 0.1 to 8%. That was when my cameraman forgot to switch my cue cards. But yes, 0.1% to 8% of the photons delivered by sunlight is what's going to be used. So that's quite a little bit. That's quite a low percentage. So that is why the plants are, the leaf is so well designed in terms of that funneling, which we'll get to. But we're still on light. I mustn't jump ahead. All right. So we understand that light can behave as a wave, as a particle. We're going to stay with it, stay thinking of it as a wave. And we're also going to consider this idea that, and remember that not all of the light that gets to the plant is used in photosynthesis. In fact, only between 0.1 and 8% of the energy that is sent to the plant, or that the plant comes in contact, is actually used as photosynthesis. The rest is reflected. It's reflected back into the atmosphere, some of green color for most plants. All right, so we have to understand the spectrum, this visible spectrum, and this type of radiation in general. I don't care too much about this, but this electromagnetic spectrum, this is very important. This section here is very important, from 400 nanometers to 700 nanometers. This is the visible spectrum. Now, this is what we can see. Now, plants are getting exposed to much more than that, but this is between 400 and 700. It tends to be what a plant can actually utilize, and that's the visible region. So what you need to understand is this this equation is Einstein's equation, energy of a photon. Remember, Einstein's that very clever dude, crazy hair, calculated this equation, and this is the equation that we use to deliver or get the energy of a photon. Do I expect you to know this equation? Absolutely not. I do not care. What I want to show you with this equation is that you see the wavelength is at the bottom. The energy is inversely proportional to the wavelength. So what I mean by that is if you put 700 nanometers down here, and you use Planck's constant and the speed of light, and you put 700 in there, you're going to get an energy, and you can actually do it. And I encourage you to take your calculators, put in Planck's constant, put in the speed of light, and put in 700. You're going to get a value. And then you do the same thing, and instead of 700, you put 400. You're going to see that 400 has a higher energy level than 700. So that's what I mean when I say inversely proportional. As this increases, as this one increases, the energy decreases. So 700 low energy, 400 high energy. And this is going to become very important when understanding pigments and understanding the different reaction complexes. So I need you to just take away from this specific slide that the energy of a photon is important. The lower the wavelength, the higher the energy of the photon. The higher wavelengths, the lower the energy of the photon. All right. Now, looking at this energy, looking at how much the plant is exposed to, and knowing that the plant only utilizes between 0.1% to 8% of light, leads us to believe that, hang on, plants actually need to have protection to avoid being damaged by high energy. Because remember, if you put shining high light on there and the energy is coming in, the plant's going to become very hot. And that's where transpiration comes in. The plant opens, releases water via transpiration, that latent heat. It gets rid of some energy, but not only that, that cuticle protects it. There are some other plants that have other alternative. There is something called chlorophyll fluorescence, which you don't have to know, but chlorophyll fluorescence essentially bounces it back, and plants are green because they're generally absorbing between the blue and purple and the yellow and red. Now, can you guys think of another mechanism or something else that plants have that could form a protection against excess sunlight? Think about it, consider it, come up with some ideas, let me know on Discord, let me know on Moodle chat, send me an email, whatever you want to do. What do you think are the protective mechanism that plants have against high light? So, some terminology that you need to just bear in mind when we're talking about light is this photon flux density or photon irradiance, and that is simply what light is being shone, how much photons is a plant receiving, what is the quantum yield, what is the quantum yield being received by plants, but more importantly, what is the quantum yield that allows for a high carbon dioxide assimilation rate. Now remember, don't forget, I said it previously, and I'm going to keep on saying it throughout this set of lectures. Carbon assimilation, carbon fixation, carbon being formed into glucose, same thing. So what is the quantum yield? What is that ideal wavelength that is going to give us a high CO2 assimilation rate? Carbon assimilation, carbon fixation rates. That is the language that we use when discussing photosynthesis, etc., etc. Okay, remember, we're talking about sunlight, we're talking about excess sunlight, some of the protection that I've asked you to go and look through. Not only does that intercellular air spaces play a role in allowing for oxygen and carbon dioxide to come in and out and diffuse and all of that, but it also acts as a sieve effect. It also acts as a scattering effect. And this is a protection mechanism to protect the plant from overheating, which we'll look at a little bit later. Okay. So, here again, this is just reiterating the fact that plants are exposed to high energy. Look at that solar output. Look at what but remember down here, wavelength, 400 to 700, that's what the plant can use. Look at the range at that solar output. Look at the energy at Earth's surface level. And this is the absorption of chlorophyll. And you can see there's two beautiful peaks over here. Now remember, absorption of chlorophyll. So it works between 400 and 700. And you can see this peak here, just about 400, and this peak here at 700, and then there's a dip here. This is being reflected. This is being reflected back into the atmosphere. This is chlorophyll. This is why chlorophyll is green, because it absorbs the purple and blue, it absorbs the yellow, orange, and reds, and you will see it's being reflected back out. Now, and I'm sure you'll remember, I pointed out that not all pigments are green. There are carotenoids, which are orange, xanthophyll, which is red, which also absorbs photons. Now that's twofold. It could absorb photons for photosynthesis. Sorry, it could absorb photons for the photosynthesis process, or it can be absorbing and reflecting and getting rid of the excess energy by means of chlorophyll fluorescence. So bear that in mind. So if you see a very red plant and it's not senescence, in an atmosphere, in a region or an environment, consider the fact that that is its mechanism to protect it from the sunlight. Okay. Now we're going to go on to the chloroplast structure. Don't forget, we have cells, we have cell walls. You can see the nice cell walls, you can see the nice structure. We know that there is lignin in those cell walls, and In the cells, all those green things are chloroplasts. Look at all the beautiful chloroplasts. Apart from chloroplasts, we have mitochondria, we have nucleus, we have all of that. We have the whatever else is in an organelle, mitochondria. And yes, pause for a minute, backtrack. Plant cells have mitochondria, which means plant cells respire. There is cellular respiration that all plants do as well, which means, remember, we respire, we take in oxygen, we release carbon dioxide. It is the same for plants. Plants do respire. There is cellular respiration, but it doesn't make an influence on the atmosphere as a whole. The reason it doesn't do that is because the photosynthesis acts like a bank, and then the photosynthesis gives that mitochondria an allowance, what it needs, and then there's still a net profit. So because there's still a net profit, we don't see the effects of cellular respiration on plants. The plants don't give off carbon dioxide, they give off oxygen, because photosynthesis kind of squashes the effects of, like, it's just a rich sugar daddy giving the mitochondria what it needs to do, what it needs to do, and you can't even see the effects. But don't for 1 minute, please don't think that a plant does not respire. There is cellular respiration, there are mitochondria, it is a natural process. We just can't see the effects because the photosynthesis gives us a net profit, if you will, of oxygen as opposed to carbon dioxide. So there's beautiful cells over here with chloroplasts. Here's a nice cell wall over here, and you can see the chloroplasts. And the chloroplasts tend to generally be pressed against the cell walls, adjacent to the intercellular air spaces. And the reason for that is that's where the carbon dioxide is. It's going to be easily brought into the cell via aquapores, so that we can have a process where in the chloroplasts it comes, the carbon dioxide comes into the chloroplasts, into the stroma, into the thylakoids. Okay, so now we see in our cell over here, our chloroplasts stacked against the cell wall. There's little green chloroplasts everywhere. We have a look. This is what a chloroplast looks like in real time under an electron microscope. And you can see the stroma, the grana. This is a grana. That's a grana. That's a grana. The stroma, that liquid. Thylakoid is an individual stack. One of those will be a thylakoid. One of those will be a thylakoid. There's a thylakoid. Very beautiful, nice stroma. So you can see this is the stroma all around here. Stroma, 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 stroma. And this is a thylakoid. Okay. Okay, so we have the stroma on the outside, and we have the thylakoid. In this thylakoid, we have lumen. And you can see that yellow bit over there, this lumen over here. Now, what's going to happen is in the thylakoid membrane, you can see this little peg over here, that little peg over there, that little peg. That is where our light reactions are going to occur. On the thylakoid, in between the stroma and the lumen, on that thylakoid membrane. On that thylakoid membrane is where the light reactions are going to occur. And it is going to be in the stroma where the Calvin cycle happens. Now, just a little segue, the Calvin cycle is called by a lot of people the dark reaction. However, this is very, very not a good term, mind my English, because these processes happen at the same time. So when the light reactions are occurring, the Calvin cycle is happening. And you'll notice I'm calling it the Calvin cycle. I don't like calling it the dark cycle because it doesn't happen at night. It just doesn't need photons in order to operate. The energy is created by the light reactions, and it's the energy that is created by the light reactions that is used in the Calvin cycle. It happens at the same time. It is just the light dependent reactions and the light independent reaction, the dark reactions, the Calvin cycle. So just bear in mind that different people are going to say different terminology. They're going to use different ways of saying it. You just need to know Calvin cycle is the same as the dark cycle, is the same as the light independent cycle. So bear that in mind. Just because it's called the dark reaction does not mean it happens at night. It happens at the same time. But the light reactions happen on the thylakoid membrane between the stroma and the lumen, and the Calvin cycle happens in the stroma. Okay. So the chloroplasts and the chlorophyll are ultimately what is going to be the photosynthetically active parts, and they take the photosynthetically active quanta or photons or quantum yield and convert it into chemically usable forms. Okay. Now, again, I am just showing you what it looks nice and zoomed in. This is within the cell. Remember, here's our cells. Here's our chloroplast. In our chloroplast, we have our thylakoids, granule, stroma. On the top here, between the stroma and the lumen, which is in the chloroplast, we're going to have our light reaction occurring, and in the stroma we're going to have our Calvin cycle. So ultimately, this is the reactions over here. This is what the crux is, and the be all and end all. So not saying if you know this you'll know everything, but ultimately this is the slide that is the abstract of abstracts. So in the light reaction happening in the thylakoid membrane between the lumen and the stroma, we have water coming in. The light breaks the height. Now, this is also something that's very interesting. Remember that water has a covalent bond between the hydrogen and the oxygen. Covalent bond means that oxygen utilizes the electrons from hydrogen, making oxygen more negative and hydrogen positive. So what happens is the water comes. We are not 100% sure how it happens. This is still an enigma to us, but a process called photolysis, the splitting of the water molecule. And it is believed that the energy from the photon is used to split the water molecules, the covalent bond. And it does it so elegantly. It's almost, now, water is a very stable molecule, and what it essentially does is it's oxidizing water. It's taking those two electrons that were originally hydrogens, and it's keeping it for itself. It's releasing the oxygen's binding to another oxygen that's been randomly left, and they're forming oxygen and they're leaving back out into the atmosphere. It takes those 2 electrons, it oxidizes. We're still not sure what enzyme is strong enough to oxidize water, which is an oxidizing agent. It's very, very interesting. So remember, I said oxidizing, you taking away. So water was an oxidizing agent. It took away those 2 hydrogens electrons, and it's keeping it mainly by itself, making it negative. And it is a very strong oxidizing agent. So for whatever is causing photolysis to be a stronger oxidizing agent than oxygen, which it is named after, it must be incredibly powerful. It's still an enigma to us. We are not sure what it does. We're not sure how it does it. All we know is it takes those 2 electrons from oxygen and it leaves 2 positive hydrogen ions, and it sends those ions into the stroma, into the lumen, which we'll get to, but it releases oxygen. The oxygen goes back into the atmosphere, and now all of a sudden this light reaction has those 2 electrons, and water has been there's been photolysis, the splitting of water molecules. So now this light reaction has those 2 electrons, and it's going to go through a process, a series, and our byproducts are NADPH and ATP, with oxygen being released back into the atmosphere. So the ultimate byproducts of the light reaction, ATP, NADPH, with oxygen being released back into the atmosphere. Then NADPH and ATP get sent off to the Calvin cycle as that energy, and carbon dioxide comes in, and ultimately processed we get sugar, and then we get NADP plus and ADP plus phosphorus here, going back to the light reaction to get energy again to be formed into NADPH and ATP to come back to carbon dioxide. So the main thing you have to know before we get started on the nitty-gritty stuff of the light reactions in the Calvin cycle is that water is very strong. It's a very strong, stable molecule. So for photolysis to occur, to break, to take those electrons is an extremely powerful process that we don't understand how it happens. And as it takes those electrons, the photons in the chlorophyll excite it. And as it excites it, it gets passed along an electron transport chain all the way to the end products where we get NADPH and ATP. Those 2 are taken to the Calvin cycle and along with carbon are processed to give us our glucose molecule. And don't forget now, the oxygen that we breathe does not come from carbon dioxide. It comes from the water molecule that has been split by photolysis. Please, crystal clear, the oxygen that you and I breathe comes from the water molecule during the light reaction and not from carbon dioxide. Carbon dioxide, the whole thing gets used during the Calvin cycle. Okay. So, remember, now we understand the basics, we know what's going on. This is, yes, we're getting there. We're absolutely getting there. So now we're going to zoom in on the light reactions. We're going to understand what the light reactions are, how they work, and boy, we are going to understand it to the T. So the first thing you need to know is there are 2 photosystems. And don't worry, I will explain how it sits in the thylakoid. I'll explain how it looks in the thylakoid. But in that thylakoid, we're looking at the thylakoid membrane. There are 2 photosystems. Photosystem 1 and photosystem 2. Now, you cannot get confused. I have gotten confused. So perhaps, like I said, with water relations, you want to draw a diagram. Perhaps for photosynthesis, you want to draw the thylakoid and you want to say, okay, here's my thylakoid. The first system is photosystem 2 and then photosystem 1. So I know that that sounds confusing, but step number 1 is photosystem 2, and then we move on to photosystem 1. The reason why photosystem 1 is named photosystem 1, even though it happens second, is because it was the first one identified. It was the first one discovered. So we've got photosystem 2, then photosystem 1. So we've got 2 functional unit sites called photosystems. And their job is to ultimately it's taken that energy at photosystem 2 is where the photolysis happens, where it takes those 2 electrons from oxygen and it splits that covalent bond and it leaves 2 positive hydrogen ions and it releases the oxygen and it's got those 2. So photosystem 2's got those 2 electrons. It then absorbs energy, it excites those electrons, and then through a series of compounds that act as electron donors and acceptors called the electron transport chain, it goes down to photosystem 1 where it is re-energized, and through that re-energized state it goes down and down again through the electron transport chain, all the while releasing energy. This is just the basic summary. I promise we'll get into the nitty-gritties a little bit more, so stay with me. And then we've got an electron that's slightly energized, and it gets given to NADP. So it now becomes NADPH. And this NADPH goes off to the Calvin cycle, where it is going to use that energized electron in the Calvin cycle. Another thing that is not on this slide that should be on this slide is also in the light reactions we get ADP forming ATP. Okay, so we've got 2 photosystems, photosystem 2, photosystem 1. Photosystem 2 happens first, photosystem 1 happens second. The reason why it's called photosystem 1 and 2 is because of the way they were discovered. So even though photosystem 1 was discovered first, it happens second. It takes place second in the light reactions. We get photolysis happening. Photosystem 1 grabs those electrons, it excites the electrons, it goes down through the electron transport chain through a series of compounds, molecules, donors and acceptors. It gets to photosystem 1, it excites it again, and then it comes down just before it gets to a more stable state but still has enough energy. It gives that electron to NADP+, and NADP+ goes off to the Calvin cycle. Okay, so this is what a photosystem looks like. Okay, we're going to do this slide and then I will leave it here and then I will carry on in the next one. So we'll finish the slide. Okay, so this is what a photosystem looks like. Now remember, there are 2, photosystem 2, photosystem 1. This is what a photosystem looks like. So here is our thylakoid. This is our thylakoid membrane. This is the lumen. This is the stroma. This is the photosystem. This is that molecule, that light harvesting complex. This is the primary electron acceptor. This is the reaction center. Now this reaction center, we're going to get to it, but this reaction center can also be called now if this is photosystem 2, it can be called P680, and photosystem 1 can be called P700, and the P stands for pigment, and the 680 stands for the wavelength, and that is the wavelength that it best absorbs at. So remember, 680 is photosystem 2, and that is more energy, it absorbs at a higher photon energy rate, whereas the P700, which is photosystem 1, absorbs at a little bit of a lower energy rate. So you've got these chlorophyll. So in the thylakoid, all along the thylakoid membrane, in this light harvesting complex, think of living in a complex where you have different houses all surrounded by a single wall. All the chlorophylls are in a complex, in this light harvesting complex, and they, like you see here, in a thylakoid membrane, they could be between packets. So one light harvesting complex can have 200 to 300 chlorophylls, individual pigment molecules in this light harvesting complex. And the photon's going to come in. It's going to excite these chlorophyll pigments, and they're going to go all over the place, and they're going to go all the way down here to the special chlorophyll A molecules. Now remember, water has come in. Water has somehow been has undergone photolysis, and the electrons have been captured here. The primary electron acceptor has taken it from that oxygen, and we've got that the electrons here. These photons are going to jump, they're going to jump, they're going to jump, and then they're going to excite these electrons. And these electrons are going to be excited, and they're going to be up here, and they're going to be like bouncing off the walls. And then it's going to get passed on via the electron transport chain along here to the next photosystem. To the next photosystem, it's going to come. Now we're in photosystem 1. And now all of a sudden, this is P700. Lower energy, but our electrons have gone down. Remember, as they went down, they released energy, and I will explain to you what they do when they release that energy in a moment. But the photon, but it comes back. The photons hit again, and it comes along the chlorophylls, jumps around, comes to the special one, and it re-excites the electrons, and the electrons are re-excited, and then they come down the electron transport chain, and they give that electron to NADP, which becomes NADP+, NADPH. Okay, so I'm going to say that again because I don't want anyone to get lost or confused. We're going to keep on building. Each stage, we're going to keep on building more and more and more until you have the whole system in place. Okay, so we're in the thylakoid. Here's our stroma. Here's our lumen. This is the thylakoid membrane. We have a molecule, a protein, a light harvesting complex, which has special chlorophyll A's down at the bottom that's going to collect the energy from the other chlorophylls that are absorbing the photons, and it is going to give it to the electrons that were accepted by this primary electron. It's going to that took the electrons from oxygen. It's going to get energy. It's going to go to an increased state. It's going to get passed on to various complexes down the electron transport chain to the next photosystem, which is photosystem 1. Once it reaches photosystem 1, it's the chlorophylls in photosystem 1, which absorb at a bit of a lower energy level, will absorb, increasing the electron again, and it's going to go down the electron transport chain again. And as it does that, before it hits the bottom, so when there's still enough energy in it, it's going to give those electrons to NADP, and NADP is going into NADPH, and it is going to go off to the Calvin cycle. Now, as the electrons are coming down this electron transport chain, they are releasing energy, and I will explain why they are releasing energy and for what it is about. but I need you to understand this slide. I need you to get the concepts and understand. All right. So this is going to be the end of the first set of lectures. We are going to be on these for a while. Please take your time. Listen to it again if you need to. You need to understand these concepts. Okay, stay safe, and I'll see you in the next video.